Hi, I'm Vijay Vaswani, CEO of Omniscope. Have you ever wondered, what if we could detect and treat the very cells involved in disease? At Omniscope, we understand your immune system plays a fundamental role in your health. Indeed, by analyzing your immune cells one at a time, we generate vast amounts of data from a patient's sample. Together, we believe that this can lead us to advancing personalized cellular therapies and diagnostics. Indeed, using AI as a force for good, we believe that the massive computational advancements today can lead to an exponential advancement in healthcare. It's a real honor to be moderating today's panel. I'm interested to learn from our distinguished guests about the past, the present, and the future of healthcare. Indeed, I've been fortunate to be part of companies who have advanced healthcare, companies such as Illumina, 10x Genomics, and Siemens Healthcare. So today, in the spirit of science as we all, I'm delighted to act as your moderator at our upcoming panel. Thank you for your time and looking forward to our conversation.
Thank you everyone for joining our panel today. I believe we're going to have a very interesting discussion on immuno-oncology, the past, the present, and the future, and harnessing the immune system to advance patient care. Today, I am joined by a distinguished panel, a group of individuals who have dedicated themselves to patients and to advancing therapy and really understanding how and where we should be investing our efforts for better outcomes. So without further ado, I invite our distinguished panel to tell you a little bit about themselves and how they have proactively decided to advance patient care. Over to you, Dr. Prat.
Thank you. Good morning, good afternoon. It's a pleasure to be here today and to learn, and also at the same time explain how I see some things about the immune system and oncology. In particular, I'm a medical oncologist by training. I still visit patients once a week. I was trained here at Barcelona at Vall d'Hebron. I did my fellowship here under José Baselga's mentorship, and then I knew during residency that I needed to be involved in research. I needed to understand what was going on from a biological perspective in order to better treat patients. So that's why when I finished fellowship, I decided to go abroad. I went to the US, in particular UNC, University of North Carolina, Chapel Hill, with Professor Chuck Peru, another mentor. And I decided to spend 4 years in the lab without being far away from patients, but learning the basics of genomics. At that point, a lot of microarray data, gene expression, bioinformatics. And for me, it was a very important moment in my life because I saw another view of cancer, a view that had a lot of potential, that already was being used a little bit. The biology to guide therapy was just the beginning. So after 4 years there, I came back to Barcelona. Now it's been 10 years ago. I created my own lab at VHIO, first at Vall d'Hebron. I went back to the clinic, visiting patients, breast cancer patients in particular, once a week. I grew my lab, and after 2 years, I had the opportunity to move to hospital clinic where I am right now. First, I was the head of the medical oncology department, and now I'm director of the Cancer Institute. My lab here at EDVAPS has grown a lot, and in particular has grown a lot thanks to translational research linked to clinical trials. So I was involved in SOLTI, which is a cooperative group that runs clinical trials in breast cancer. I led the group for several years. I was running trials myself as PI, but most importantly, my lab here at EDVAPS was and is the reference lab for that group. So basically, we were running samples from patients, blood, tumors, in trials that were asking particular clinical questions. So we link all this biology to clinical endpoints. Many years of discoveries, a lot of publications, but I felt that I needed to do something else, basically, because when I see patients, I see so many questions that don't have answers. And I saw a lot of potential of many of the discoveries we were finding, but nothing was happening. So I decided to to become an entrepreneur, create a company, a spin-off company called Reveal Genomics, which is a diagnostic company in the field of oncology, in particular breast cancer. And it's been now 3 years and a half. I've learned a lot also from aspects that are important, regulatory, legal, and business. And so far, we're very happy because we already have a product, a diagnostic product that actually uses the immune system to guide therapy in early stage HER2-positive breast cancer. So as you can see, I'm involved in a little bit different pieces. Of course, I don't do that by myself. I collaborate a lot, and especially when we talk about the immune system, I think it's critical that all players come together to address all the unmet needs that hopefully we're going to discuss today.
Fantastic. Very inspirational to see science go into entrepreneurship and really make an impact. Dr. Piulats, I'd love to hear more about you.
Hi, good morning, night, afternoon, everybody, and thanks for inviting me here. I'm sure it's going to be a very interesting conversation we're going to have here. I'm Josep Maria Piulats. I'm medical oncologist at the Catalan Cancer Institute. I did my training here in the same hospital. When I finished my training, I inherited the two tumors that nobody wanted in the hospital, that were melanoma and prostate cancer. And I've been lucky because in the last 15 years, two of the tumors where they expanded more treatments that allowed me to be in contact with targeted therapies, with novel hormone therapies, diagnostics, checkpoint inhibitors, and now even T-cell engagers. I also visit patients 1.5 days a week, and I also am involved in research. Here in the Institute, I'm the head of the cancer program at IDIVELL and in the hospital at ICO. And I'm also co-leader with Ramon Alemany of the cancer immunotherapy group here at IDIVELL. And the main interest of the group is just to develop novel treatment assets. Ramon Alemany is known because of his background generating viruses. Some of them are being used in the clinic, and they have a spin-off where they're testing one of their assets in pancreatic cancer with interesting results. And here in the lab, we're moving forward in other treatments. We're currently running a clinical trial with mesenchymal stem cells infected with viruses in cold tumors, to trying to make delivery of the virus directly into the tumor, in a tumor that's not with a well-developing immune system. And also together with the sanitary sheets, the BST and VIO, we are currently conducting 2 clinical trials with adoptive cell therapy in special patients populations that they're not really frequent, but we consider that they are highly immunogenic. In that regard, we're doing 1 clinical trial in germ cell tumors that are refractory to chemotherapy. The also tumors are tumors with a lot of cancer testis antigens, and thus we consider them that they could be a good target for that. And then the other, it's a special group of low prevalent diseases that have something in common, that it's monogenic diseases with defects in SMARCA4, SMARCB1. There tends to be young people with kind of like rhabdoid tumors. We just filed for another clinical trial with a novel T-cell isolation protocol, adding several drugs that we've demonstrated in vitro that increase the T-cell reactivity. And we have to do that going the hand with people who know how to develop technology to follow up those patients. So not all the patients are going to get a benefit of those therapies, and we have to try to identify them, and all of the other patients, they're going to be good responders, and we have to be able to identify them and even learn from them, and even harness that immune system, like isolating the TCR and expand those treatments to other patients. And that's what we want to do here in our group.
I think it's fascinating for our audience as well to see that you're participating in these groundbreaking clinical trials but you are still seeing patients. And I think that's something which, for our audience, this requires a lot of time and dedication. And it also means that learning from the patients, Dr. Piulats, means that you are enriching the actual ability to cure as you actually test out these new groundbreaking therapies. So thank you very much for that. Dr. Almaguel, I know that you're joining us all the way from California today, and I'm very excited to hear about your work.
Thank you for the invitation. If we go back to why I'm here today, it's because my passion for patients, right? When I decided to go through the hard, narrow path of medical science, I mean, MD degree plus the rigorous time in the laboratory, right? To get that foundation to do something in the future, because I did not want to follow the cookbook. I wanted to create a cookbook when there is a need. And unfortunately, there is a lot of need, especially in oncology. And after radiology, nuclear medicine, and had the opportunity to dive into oncology and to diagnostics, and I'm in a position now where, yes, I see patients in clinic a couple days a week, but I see patients every single day. I am involved with patients at the same time, full-time laboratory, multiple clinical trials in this field, and you can't do that alone. So you have a team and you have collaborations. I'm working in academia and private practice, trying to create a national network so we can get the best treatments for patients now. And there is such beautiful opportunity to create a multidisciplinary program that we could actually take a patient from no hope to in the future use the most rare word in oncology, which is cure, right? And that's why we're here. The rest is details.
Dr. Almaguel, I think it's incredible to actually hear somebody say we need to redesign a cookbook and go to cure. Can you tell us a little bit more about the role of nuclear medicine in this field and how you see that in Boston?
The idea of nuclear medicine coming from unclear medicine to clear medicine to nuclear medicine is that in the past we had tests that was very hard to work with. Now, with the advances in PET-CT, we're able to see beyond our wildest dreams, right? I mean, I can see a PSMA PET-CT showing this tiny metastasis that in the past, would have put a patient into very aggressive therapies with lots of side effects, and now we're able to address that dot that is causing the recurrence. So the idea is, in the future, I believe that every cancer is going to have its biomarker. We're going to have targeted therapies. We will not see what we're doing today of doing therapies that are not, we don't have a direct way of monitoring, right? So in the future, I see a future where there is no more breast cancer, prostate cancer, melanoma. It'll be like PSMA positive cancer, HER2 positive cancers, and then you just have direct treatment for those. And you'll be crazy to think that we had to take a huge piece of somebody's body, because that's the only way we think about helping that patient live longer. And if you don't get excited about that, I mean, I can't tell you anything else. I mean, I think the future is bright, and every single step that we're taking today might not look like we're making a huge change, right? Because even though I'm seeing change, right, but there's gonna come a point where the next step is the gap of the Grand Canyon, right? Just all these steps by many people together suddenly will click, and they say, "Oh, there is oncology, imaging, radiation. If we put all that together, we can change the field." And I don't know how many of you have been into funerals for your patients or to birthdays, but I can tell you it's a real thing. And if somebody says that we're done everything, there's nothing else to do, then we wouldn't be in this panel, right? There is a lot more to do. We still have thousands of patients dying every year, right? I mean, I think last time I checked, more than 600,000 patients dying just of cancer, right? There's so much need that we cannot be just happy with what we have accomplished. And there is a lot more people that we need to help, and that's why we need to put our heads together and look forward, right? Based on the foundations that we have been blessed to be around. And
I think
let somebody else talk now.
That's very sobering, and I think it brings us to exactly why all of you have dedicated so much time. Dr. Heyn, how did you get involved in this area? What's your genesis?
Yeah, I think I'm here because I love technology, and not for the sake of developing technology, but really to see that applied on clinical or to solve clinical problems and benefit patients. That's why I co-founded Omniscope. That's why I also work as a group leader at the CNAG, the Spanish National Center for Genomic Analysis. My background is on genomics. So I've seen the field evolving over time. I've been working on single cell and spatial genomics over the last 10 years. And really the teams I build or companies I build is a hybrid between the technology part and the computation part, biology part, now much more AI being involved. And I've seen especially the field of single cell genomics moving from profiling really single cells, being sorted in plates, to profiling thousands of cells and really contributing to solving or developing insights into complex problems like tissue microenvironments, inflammatory diseases, immuno-oncology. And I think what we've done over the years is contributing to the technology part, benchmarking technologies, co-developing technologies, mainly the area of our contribution to the Human Cell Atlas project, which is a huge international initiative really to decipher the complexity of the human and really to build a blueprint, a model of the healthy human system. So we have been working a lot on the technology part. It's also my role as a chair of the technology working group of the HCA, but we also contributed to the immunology part of it, so the Immune Cell Atlas, recently publishing the Tonsil Atlas, describing the complexity of immune cells in a secondary lymphoid organ, the tonsil. But we're now taking it further to profile diseases like inflammatory diseases. We have a lot of clinical trials ongoing using single cell, now more and more spatial technologies in the area of immuno-oncology, teaming up with clinicians, teaming up with pharma, really to helping explain response to checkpoint inhibition, to quantify response, to track response in blood. So we've developed with Omniscope technology that goes very deep, that provides high throughput insights into immune cell action in cell circulation. So we use that as a live tracking tool. And that really helped us to develop a framework on the technology side, to track cells, but then also to learn the rules behind the patient responding to checkpoint inhibition. And that hopefully in the future allows us to stratify patient. So not only to live track the patient response in real time, but really also to stratify patient before a time point of diagnosis, and then to tailor the right treatments. So I think a common theme is really to use the immune system as a sensor for treatment response, but we can imagine that being applied in early diagnostic disease follow-up, and really to make personalized medicine in reality by using the patient's own immune system as sensor and to monitor that in real time using high-throughput, high-resolution technologies.
Fantastic. I think, as Dr. Almaguel was alluding, it's going to take all of us collectively efforts to actually advance outcomes for patients. I think as we now sort of get into the main part of our session today, it would be really useful to understand how each one of you sees the past. A lot of you have worked with luminaries in the field, and you've alluded to many therapies and many discoveries which have been groundbreaking and have advanced healthcare. But I would also assume that many of these past achievements have also left some unmet clinical needs, which is why what I hear from many of you is that you are actively in the present looking to actually address some of these. And of course, as we come towards the tail end of our session, it will be very, very interesting to hear from all of you who are in contact both with patients, but also with the advances in diagnostics and in therapies, what you see as the future. What are reasons to be hopeful, and what are the challenges in the future which we will have to prepare? But perhaps we can kick off with understanding where we came from. And Dr. Piulats, perhaps you can give us some insights on what you have seen in the past and the challenges, and today, what you see as abilities and strategies to address some of the gaps.
So I think we are here because of the understanding that the tumor has different ways to evolve and defend itself from the immune system. And one of that is the expression of a protein on the surface, that's PD-L1, and that interact with a protein in the immune system, that's PD-1, and that silences the immune system so the tumor is protected. So one of the problems is that until that moment, we've always talked about immunotherapy, and we were talking about a kind of like a broad immunotherapy that was inducing many changes, like putting high dose IL-2, et cetera, and this kind of novel immunotherapy is something completely different, with giving cytokines at high dose, et cetera. We didn't know exactly what we were doing. Probably we were just breaking balances, and probably the balance was going to the good side, but it could also go to the bad side. Here, we're tackling just one protein, so we're doing really a targeted therapy in the immune system. So that made so which patients are going to be getting a benefit from that treatment? So probably the patients whose immune system is suppressed mainly because this protein is active. If that's not active, the immune system is not going to work. So no, the immune therapy is not going to work. If that's active, but it's not the main driver of that immune protection, the treatment is not going to work neither. And that's something that seems like really clear and like that, like ABC, like 2 = 4. So we did not have that clear for many years, and we've been treating tumors that now we realize that we should not have been treated, and probably they've been treated wrongly, with not a good selection. So I think one of the problems also, it's been that before the immunotherapy, we had the development of the target therapy. And a lot of the things, how we started to understand how the treatment of cancer was with target therapy, we translated all those thoughts to immunotherapy, and probably immunotherapy is a completely different thing. So it's kind of like within target therapy, we know that the patient, if they don't have this mutation, this amplification, this fusion gene, the treatment is not going to work. And we have really good biomarkers to detect which patients are going to get a clear benefit on specific drug. And we wanted to do that exactly the same with immunotherapy, but immunotherapy, the immune system is much more complex, and it's harder to understand, it's harder to study, and also it's dynamic. If you have a mutation, you're always going to have that mutation. Probably with the treatment, you're going to acquire another alteration that make that treatment resistant. So it's really easy, compared to the immune system. I'm not saying it's easy, but compared to the immune system, it's easier, to see that, And then suddenly we have a completely different thing. So we know that there's heterogeneity between lesions, and even in the same lesion, depending on where you are getting the biopsy, you can have a completely different result, etc., etc. And we, as I said, in the first trials, we didn't put all that complexity in context. No. In fact, 10 years ago, when I was doing talks about biomarkers, I was saying no, and we're discussing PD-L1 and which antibody we should use. No, there were like 4, and some of them had completely different results. And now we have to look at it in the immune system, and now we have to look at the cancer cell. No, so it was all that mess that like self-imposed mess that we generated, and we can discuss why that happened, and now we are almost like kind of like forgetting about all that, and in those speeches like 10 years ago, I was saying no. So we were trying to use those to select the patients that were going to be treated versus the patients that we were not going to be treated. And I said no. So these biomarkers should help us to decide which patients are going to get a benefit of the monotherapy, and the patients who do not have an effect with the monotherapy we will need combinations. And all these biomarkers should be really not to decide we're not going to treat that patient. No, it's kind of like how can we personalize those treatments even more? No, and in that regard, I think we're getting now there. We are starting to understand better why some tumors, like for example, I'm working prostate cancer immunotherapy, talking immunotherapy as PD-1, PD-L1. No, it's not working. No, so what do we have to do? No, we're getting better information about that, and I am probably the next waves of clinical trials. No, with novel treatments, novel treatment modalities, novel antibodies, T-cell engagers, adaptive therapies, etc., advanced therapies. No, so I think we have more tools, but that also means that we need to understand better, the immune system.
And I think that's why we're so excited with the new technologies being available and being applied and yet in clinical trials, and not yet in clinical practice, but really in a clinical context. Because in the past, we've seen that next generation or back-end microarray technologies were useful to work on the DNA of cells, more near the tumor fraction, really to identify driver genes, tumor heterogeneity, and then specific treatment, like targeted treatment for BRAF or EGFR. But technologies were useful in a sense, generating a bulk profile of a tumor, but they were not useful enough, I think, for the new therapies that came up, checkpoint inhibition, because we're not able, using bulk TCR sequencing, using bulk RNA, bulk DNA sequencing, really to decipher, disentangle especially the immune components of a tumor. And that's why we lack biomarkers to predict the outcome of checkpoint inhibition. I think that's our excitement with these new technologies being applied really to disentangle immune cell fractions, plasticity, immune cell states, and when really to put this into a clinical context, correlating that with patient outcome. And really, for me, this is a second generation of biomarkers that we're now developing that go beyond this bulk profiling of tumors, but really to have a high resolution profile of cells in tumors and tumor microenvironments, with new biomarkers coming up that relate to cellular states, and cellular states being susceptible in the end for checkpoint inhibition, being susceptible to be reactivated. I think that's what we are doing already, but I think this is the second generation of biomarkers moving forward.
It really sounds like we have a gap, which has been a product of the past, between advancements in therapies and understanding which patients would actually benefit, and being able to have this generation of biomarkers which can allow us to make these clinical decisions. Dr. Prat, Dr. Almaguel, what do you see as the past and the current challenges?
I mean, I will defer to the medical oncologists in the room, but I believe that the main problem from the past was the lack of precision. Let's give you this medication and see what happens in 3 months when we do the imaging. I think now we're understanding that that was wrong, and even though that was all I have. Like I gotta tell you something, right? I'm presenting and I'm showing these pictures, right? And my colleague will say, "Hey, if you had shown me those pictures 10 years ago, I would have treated this patient a lot different." And it's a fact, right? Uh, we need to move to more precise therapies and to better decision making. And this community of working together is unique, right? Because I have patients say, "I want this therapy because it's the best," and even though you think it might not work because I don't see the pictures, right? I don't see he doesn't have uptake for the biomarker that I'm gonna treat him with. So I say, look, we need to find something that is for you better, because if I am treating you with something that is not gonna help you, trust me, I can always make it worse. If I can make it better, I can always make it worse. So the idea of precision, and I can tell you, the need of precision is key, because if we were able, if we knew what we know now, I mean, if you go back, there are patients that you know that if you had this technology now, you could have saved them. And you cannot travel through time, but we're trying to prevent that in the future. That's why we're working together. That's why we put it all our hearts together, right? In the future, there'll be a complete new field where using low-level PD-L1 or in combination with targeted therapies like in Diagnostics, patients will respond better. Or giving some radiation before the PD-L1 inhibitor might change the way we do things. All those things are being evaluated now. The idea from, if I can take a leap from the past, is that we are in the future where there will be more collaboration and more precision.
Yeah, I think precision oncology is the word. And when we talk about precision oncology, I mean, that means that we need precise drugs and precise therapies, but we need precise diagnostics, right? I think here is where also we're seeing a gap, no? And the therapeutic part is evolving very fast, very precise, very novel drugs, right? But from the diagnostic point of view, the technology is there, no? As Dr. Heyn pointed out, technology, Omniscope has technology, but how do we apply this together with therapeutics, no? And here there's a gap that we can discuss why, no? But just to give you, especially for the audience, a view of how things have changed, in particular the place where I am involved more, which is especially breast cancer, in particular early stage breast cancer, right? And in the last 20 years, this has changed a lot. No, breast cancer was one disease 20 years ago. Thanks to translational research, studying tumor cell features, we started to subdivide breast cancer into different groups. And this is not just a scientific exercise. These groups have important meaning in the clinic. It means that patients will be treated differently with different drugs, and this has been thanks to genomics of the tumor cell level. And we're using this today. And thanks to that, 20 years ago, we were giving chemotherapy to prevent a metastasis in all patients with more than 1 centimeter tumor. That was the criteria. So everybody went through chemotherapy, most of them. Now we are tailoring more the use of chemo. But at the same time, it is not enough. I think you all brought it up, right? Even in breast cancer, that the cure rates are very high, it is not enough. And I think in the last years, especially the last decade, what we're seeing is that the immune system here has a lot to say. In breast cancer, a tumor that in general, now we have immunotherapy for triple negative, but it has taken a while to have anti-PD-1, anti-PD-L1 therapy approved. But we know, and thanks to translational research, we've learned, right, thanks to technologies, that the immune system plays a role, not only from a therapeutic point of view, but also to understand the behavior of that cancer. And we've learned that by studying the immune component right there next to the tumor, we can predict things like overall survival of patients, if the patients will respond to a therapy or not. And I think this is happening as we speak. And it's happening with simple gene expression data, and not even complex technology and high throughput technology like the one Omniscope has. So even with gene expression data, for example, we have developed this test called HER2DX. It's the first tool that integrates gene expression data from tumor cells, but also from the immune microenvironment, specifically B cells and plasma cells. And with that, we gain prediction to predict things like if the patient will achieve what we call pathological complete response, which basically means that the tumor will disappear if this patient receives 3 to 6 months of anti-HER2 therapy, and disappearance of the tumor, literally. So the surgeon will go and do surgery, and it's going to be a scar, what's left, right? And we can predict outcome, and this has clinical utility. So, and this is just the beginning. So when we talk about the future later, this is just the beginning. If the immune system by bulk RNA expression can already help us predict that, I cannot imagine what TCR, BCR, spatial profiling will be able to tell us to these patients. Because again, I'm in the clinic and I see many patients being overtreated, and I know what that means for patients receiving 6 months of chemotherapy, and not only during those 6 months, but also long term. And also immunotherapy. Now that we have anti-PD-1 therapy for triple negatives, it's a difficult therapy to give. Many patients have toxicities, important toxicities, lifelong lasting toxicities. So I think here the immune system will play a role. And let me just finish with this, and we can discuss this, because I do think that the technology is there, but the question is how we show clinical utility of this technology. How we build the evidence, just like the drug industry is bringing so much evidence, level 1A evidence for new therapies, and these get approved, even if you treat 10 patients only to benefit 2. That's a current situation in general in oncology. But how do we bring that evidence for diagnostics? And I do think that we need to run trials, we need to run prospective studies with these diagnostic tools in order to bring that evidence. If not, I think we're going to have amazing technology, amazing ways to look at cancer and the immune system, but without the proper evidence to be used in the clinic. And I think here is where diagnostic industry, academics, and potentially also pharma that might be interested in particular settings, we need to build the trials with that diagnostics. If we don't do that, I think we're gonna fail to patients.
I think it's really interesting to hear all of you collectively bring up some themes with achievements in the past, but some of the challenges. Essentially, we are in an arms race between the immune system and the tumor. And obviously, there's the elements of immune evasion, immune escape, the recurrent theme of personalization and actually understanding the dynamic within a patient, particularly on the diagnostic side, as has been pointed out by Dr. Prat, seems to be something which we really need. And another theme which comes out of this discussion is the incredible advancement in tools, whether they are on the genomic side, the spatial side, the immune side, but all of these tools which seem to be able to give us those insights into harnessing the immune system, which seems to be so fundamental to all of your work, comes up. And indeed, the participation of all of the various stakeholders from pharma to the clinic in order not to fail the patient, because at the end of the day, what I hear from all of you is a deep driving desire to benefit the human condition and the human on the other side. So I think this was a great way for our audience to actually hear the many elements from the past. Before we go to the very exciting future, let's get into the middle of our conversation and really try to understand just a few of the elements, maybe the 2 or 3 main things that you see in the present. What is it that you are doing today, and of course with your collective teams, which can actually improve the outcomes? And perhaps, Dr. Heyn, we can start with you, where you have a view as a technologist. What is it that we are getting done today? What is it that we can look forward to, which is already being put into the real world?
Yeah, and I can also quickly comment that it's a good segue anyway, that the technology being applied today, what Aleix was saying about biomarker development and product development, the diagnostic side. Because in the end, what we do at this stage, I think it's still we're in the discovery phase, where we apply a lot of technologies to get a, let's say, 360 view of a tumor, of the tumor microenvironment, profiling the tumor, profiling the blood, having the longitudinal aspect. So we have a spatial temporal overview of immune cell activity on a system biology level. But obviously, to develop that into a biomarker, into a single biomarker, or into a single diagnostic product, there's a long way. But for me, at the beginning, is always the understanding that we need to understand how immune cells interact, how immune cells are trained, how we can track them, how they enter and kill tumors in the end. And what we're doing, for example, in clinical trials to kind of generate this full overview. For example, in colorectal cancer, we profile the tumor microenvironment, and then we track that over time. So we develop a high resolution, for example, immune repertoire profiles from the blood. The patient gets checkpoint inhibition. We track that over time. We quantify the response. And then we look what immune cells are infiltrating a tumor, what makes a responder. So it's kind of to get a mechanistic insight, but already applying tools that can later convert into biomarkers. For example, the immune setting profiled through a biopsy of a tumor, but also the immune setting in the blood, the immune repertoire being fit for purpose, being kind of fit to be reactivated. At the moment, we are live tracking that, but eventually we'll go to type 1.0 and really see if this can be turned into biomarker for patient response, for checkpoint inhibition, for tumors, for cancer. But again, we come back to this sensor element. The immune system is very universal by definition. This can be turned into different directions.
Well, I completely agree. And if I would explain how I see the present, I see it like an indigestion of data. During those 10 years, clinical trials have generated a lot of data, and they've been focusing mostly into aspects, into biomarkers that at the end, they are not really looking directly to the problem. They're just surrogates of something, TMB, for example, that it's a surrogate of immunogenicity, antigenicity, and then PD-L1, for example, that's a surrogate of immunogenicity. But as we said, this is not as simple as that. And those biomarkers, the only thing they're useful is to concentrate a pool of patients that are enriched with responders, but there's non-responders in one side and responders in the other. So now I think it's a moment that we have, and then all those biomarkers that enrich, when you get to the combinations, they lose completely the power even to concentrate patients. So we're in a moment that I think we need to see what we've done and see where we want to go. And of course, it's a moment to collaborate also, because I think there's a lot of samples from a lot of patients treated. They're just stored in biobanks from different companies or institutes with really good clinical information, and some of those old and not really good biomarkers, information. And I think it's a moment just to, as I said, to try to see what it should be next, and try to pioneer in this field as you're doing in Omniscope. And as I said, collaborate with all these institutions, with all these clinicians who are really enthusiastic about this field, that they've demonstrated already. They're active, trying to getting all this data, generating all these questions, trying to answer all those questions, and move forward from there.
Yeah, I mean, if I can follow on that, fully agree with the two aspects. I think right now the wealth of data that has been produced is amazing at all levels, even from very precious samples from clinical trials. So that's there. I think a main theme right now is to stop for a second and ask ourselves, what is the clinical question that we want to answer? And I think we are lacking a little bit of vision here, because there are many clinical questions that can be answered. We see a lot of patients. There are many aspects. We want to answer who's cured versus not in one particular setting, who needs this and who needs that. A biomarker that prevents, for example, or that talks about if the patient will develop toxicity to a particular therapy. I mean, there are many clinical questions with different aspects, different areas, different. So I think defining the clinical question is one aspect, and then use all this data that many times have already been produced, is sitting there, the samples or the data is already produced, and to use that data to try to answer that clinical question. And I think if we don't do that, again, we're going to fail to patients, because sometimes we look at the future with novel therapies, which is fine, and we're going to talk about the future, no doubt, it's bright, but we already have so much to do with what we have. But defining the clinical question, I think, is going to be critical. And it's going to be, we cannot expect that we're going to identify one feature, one combination of features that is going to help us, in all aspects of breast cancer, for example. Most likely, we're going to be in a situation where each different feature will help us in particular questions. So this brings complexity, no doubt, but that's what it is. Another aspect, no doubt, is collaboration. I fully agree. I do think that here academia needs to play a role. Diagnostic companies, pharma companies, also regulators, I do think they need to come in, because if not, again, if we're going to be only accessing retrospective data from clinical trials, it's fine, and we can develop biomarkers based on that, but we need to be able to run prospective trials. And I think we are in a situation where prospective trials are starting to be run with complex diagnostics, not just with simple immunohistochemistry. We are already there, and it's happening, but I think it's still a minority of the cases. So I do think that if we all see, what is the clinical question, how are we going to achieve, with what drug, what particular setting, with what diagnostics, and also regulators help here, even with funding. At the same time, I do think that we can do that, and it's happening as we speak.
And I agree. The present is we have a lot of data, we have access to new technology, but we need to define how do we use that in the most efficient and precise way. And that's where collaboration comes, and understanding that we need tools to digest that data and to apply it to patients.
I think it's been most refreshing, even for our audience, to hear some of the issues. And I think Dr. Piulats really touched on something very important, that biobanking and actually using those samples for utility is absolutely critical. In a way, it's a way of using the past to future-proof for what may be happening. And I entirely echo Dr. Prat's sentiment that we're going to need a lot of different stakeholders, including the regulators, to actually come in and say, we need to better guide the intervention based on understanding the patient's dynamic. Indeed, the element of collaboration, I think, is one which all of you really embody, because you have collaborated in so many different roles. And as Dr. Heyn was alluding to, to answer a clinical question, one needs technologies which see enough, whether or not, for example, that cancer is indeed expressing the target, so that we can line that up to the patient, as is the case of Reveal Genomics, or what are the patient immune dynamics in response to therapy, but also what Dr. Heyn really referred to, which is, what is it a priori? What does that immune setting look like before we even start to treat a patient? Dr. Almaguel eloquently sort of summed it up when he said, maybe 10 years from now, we look at what we've been doing and we might feel, was that the right thing to do? But of course, the benefit of hindsight is always useful. I guess this is a really great way to go into probably what our audience is most also interested in, which is the future. And I can't but help ask you a few questions as you sort of start to address this. I guess, we've talked about combination therapies and the ability to put therapies together to beat cancer. We've talked about the large amounts of data and how do we use that correctly. So obviously we are living in an area which is pertinent to all of us, the revolution happening in artificial intelligence, which will hopefully allow us to collaborate and to gain far more clinical insights into these large repositories of data, many of which are coming out of trials. But by the same token, we have new technologies which can generate vast amounts of data from existing retrospective patient samples, so that can also provide us the insights. So as we go through the vision of the future, which you are aptly positioned to speak about, I think our audience would also be interesting to see how you view the role of AI as a force for good. We've talked about resistance to therapy, but I think we should also maybe talk about the resistance within the medical sector to certain new technologies. Sometimes, despite the fact that we are all in science, we sometimes lag behind other industries in picking up these technologies, which can make things better. And I'm also very, very curious to hear from all of you, particularly in your domain areas of expertise, what are the things in the future which we need to be preparing for? What is it which we're seeing, for example, in nuclear medicine, which can change the status quo, for instance, in prostate cancer? What are we seeing as areas in breast cancer, for example? Dr. Prat, you mentioned early detection, which I'm really glad you did, because if we can tackle the problem early on, perhaps we can mitigate the suffering that occurs in later stage diseases. So I'm hoping that you can also give our audience a bit of a feel of how you see these technologies, these therapies, and why they should actually be hopeful for the future of healthcare. Maybe if it's okay, we can kick it off with Dr. Prat, since you've already touched base on a few of these elements.
Well, so my thing to talk about, and I'm sure my colleagues will bring different aspects. I think that the future is exciting. I am very excited, and I think here in front of us we have a very heterogeneous audience. And I think, and I would tell you that please be involved and in this effort. In many aspects, we're going to need a lot of people and with different backgrounds involved in this effort. My vision is that the technology is there. So my vision is that we're going to be testing continuously the value of biology for clinical care. And what I mean by that is that right now in the present, we are testing the value of drugs and clinical trials, especially in large cancer centers such as ECO or ours. Continuously, we're putting patients into trials, testing new drugs, and this is happening as we speak, and we're learning, and some drugs get approved, some combinations, some do not, and this is our routine, and this is going to be even potentiated even more. But I envision diagnostics being part of that. In other words, running trials where we're testing the value of diagnostics, where we're testing how diagnostics can answer clinical questions, and we're doing that as we run a clinical trial for a drug versus drug B versus drug A. We do the same for diagnostics. I envision that, because if we don't do that, I think we're gonna, again, as I said before, fail patients. The technology is there. We can do these prospective trials. It's just that we need to do it. So I do think that's gonna happen. At the same time, I envision that we need to work all together, and also we need to make, let's say, life easy to patients and physicians. What I mean by that is that this is very complex information. AI is complex. Understanding the immune system is complex. Much of these potential biomarkers are very complex, but at the end of the day in the clinic, we make binary decisions. We treat, we don't treat. No, I change therapy, I don't. I combine it or not, right? In particular settings, right? And we need to be able to provide to physicians and patients that right information, that they can make the treatment decision at that point. And it's going to be binary, that decision. So what we build upon AI and immune system and so on needs to go into that direction. Because if not, we're going to be left with a lot of information and physicians completely lost about what to do, even if you build evidence around it. So I think also understanding also the patient and physician perspective, not the physician, maybe there's a clinical scientist that understands the biology, but the physician that is seeing patients every day, 5 days a week, right? Even sometimes more than one cancer type. they need help in order to integrate this complex biology into the clinical setting. So there's a gap there that I think in the coming years we need to put a lot of effort. So that's my first comment, but I'm sure others have others.
I would fully agree with Aleix that futures is already happening. We're coming back from ACR. I think we've seen a lot of new therapies coming up, being tested in clinic trials. And there's a lot of development in next generation checkpoint inhibitors beyond CTLA-4, PD-1, PD-L1. There are new CAR-T designs being tested that even go beyond this surface and recognition compartment, but really do kind of intrinsic tuning of CAR-Ts to make them more persistent, make them more active and more precise. And talking about ADCs, bispecifics, I think there's much more coming on the kind of target discovery that we will be much more precise. But I'm particularly excited about the kind of personalized part of it coming from the cancer vaccine side, coming from the TCR-based T-cell therapies. So I think there are a lot of successful trials already being done, and I think it is really then the smart design of a vaccine that is personalized based on the neoantigens, so the mutations being presented by a tumor cell, or buying engineered T cells with TCRs that are proven to recognize the tumor and to have them maybe as a second-line treatment after immunotherapy change. But I think the future, the ultimate goal is to have kind of off-the-shelf drugs where we have common epitopes being used in cancer vaccines, like KRAS hotspot mutations, being common TCRs being reactive against cancer with a given background. And I think this is where then we move to these off-the-shelf therapies that are better scalable, that are much easier to manufacture, much kind of cost efficient to produce. And I think to get there, we really need to generate large data sets. And Vijay, as you mentioned, AI is going to be at the core. So we have to be smart in generating data, the right data, high quality data, and then to leverage latest AI technology really to learn the rules behind T-cell recognition of a tumor cell. And we need to start cracking that immune code to make a T-cell and a TCR receptor into kind of tune that into more universal off-the-shelf.
I fully agree with everything you said, no? And I think during the other interventions, no, we've talked about the classification of the patients, et cetera, et cetera. And one thing we have to move forward, no, is to do that also, no? And in the field of breast cancer, no, thanks to doctors like Dr. Prat here present, and no, and other tumors like lung cancer, we have good classification. But even though they have a good classification, you hear how they're complaining that you're not getting everywhere. So imagine other tumors where we do not have that, no? For example, prostate cancer, no? That everybody's treated almost the same way, except a real small pool of patients that have HRD mutations. No, we can just now from ACR, I presented the results from a clinical trial where we've been treating patients with PARP inhibitors, and we found that a non-HRD group of patients that suppose like 35% of all the patients are getting a benefit are the patients that have the fusion gene between 10%, no? And those are a big, important group of tumors, and probably, no? And now we have to validate that prospectively, but those patients are getting, they're doubling the PFS, no? From 6 months to 12, for example, no? So we have here something that we were not expecting, no? And no, thanks to all the molecular analysis done, et cetera, we've done those retrospective, in that case, analysis, and we found this group that it's even bigger than the HRD, no, related, no. So that's what I'm saying, is that we have to learn to classify better the diseases. And here I would like to talk also about a big group of patients that we're not talking that much, that the rare tumors, for example, no. So the rare tumors are not that rare, no? So 30%, 1/3 of all the patients with cancer have low prevalent cancer. So it's not rare to have a rare tumor, no? It's rare, no? But they're really hard to approach, no? Because they're scattered. Sometimes they're not visited in referral centers. no, et cetera, et cetera, et cetera. No, there's no big biobanks from the, and sometimes those diseases tend to be more homogeneous. No, like they're not that those many subgroups. One of the rare tumors I'm working with is uveal melanoma. We see that 100% of the tumors have a mutation in one axis. No, and we don't have a target therapy to block that axis, the GQ11, but 100% of the cancers have that. No, imagine if we could have a treatment for that, or here in the lab, we're working with TCR, trying to identify TCRs against those mutations. No, and here's all the technology done in collaboration with the CNAG and Omniscope. We're analyzing patients right now to trying to find enrich T cells that have been exposed to APCs pulsed with the peptides. No, try to enrich and try to find TCR that we can use on the shelf to treat patients with those mutations that in that specific tumor that are really frequent. And we are also even moving to try to generate a vaccine, RNA vaccine, just codifying those. And we know now that in the cancer vaccine field, there's several companies that they're trying to do neoantigen approaches, but probably that's not going to be just for neoantigens. There's other antigens that they are not coming from mutations that we know they're highly antigenic, and they could be also included in all the on-the-shelf vaccine in that regard. No, and all those new technologies that allow to track TCRs in the tumor and also in peripheral blood or even in urine in the case of bladder cancer, et cetera, they are going to be really helpful. And as a sub-product, we can even find TCRs that are specific to a specific cancer antigen that we can even isolate, clone, and use to generate novel therapies. So I think we're in a really interesting moment, and what we do right now it's going to define the future for sure. And I'm I'm an enthusiastic of the oncology in that regard. Sometimes your colleagues the typical questions, if you were doing medicine again, what would you do? I'm one of those that says that I would do exactly what I'm doing right now. And I like the moment that I am right now. The moment that all the things that are being developed. And I think we have a bright future. And talking about AA, I think we just have to embrace it. We have to develop it in a rational way, but we cannot close our eyes to that. We have really huge, humongous amounts of data. And we'll need tools, and AI is a tool for that, to try to interpret all that data.
I feel our future visions are very much aligned, Professor Madi. Very much looking forward to kind of put the building blocks together.
That's it.
And I was gonna say that I'm excited about the future because we can integrate multiple fields and we can work together. And I know that together we can do something. If I tell you that I'm going to do it myself, it's impossible. It takes a team and it takes an understanding that you need help. Every time I get an amazing response, I have to say, oh man, this is great, but what about the other 100,000 patients that I can't get to? If we had detected this 10 years ago when the cancer was little, then we probably have a better chance of a best outcome. So it's complicated, but at the end of the day, it is what it is. We need to work together. We need to harness the technology, and we need to embrace change.
And Frankis, maybe one follow-up question, because the precise technology that you're describing, how do you see the role of the immune system as in the end, it's a precision medicine, precision therapy tool? In clearing the tumor and avoiding relapse? Do you think it's a one technology that solves it all, or it has to be a combination of activating the immune system to avoid long-term relapse or the tumor?
It's a combination. There is no question. Actually, without the immune system, none of these therapies work, because you damage that DNA, but the cell can try to repair it. So if you don't have the immune system identifying those cells that are a problem, we know that there is just we don't have long-lasting responses. So I have patients with great responses, then something happened in their life, a lot of stress, and cancer comes back. The patient with the best outcomes are the patients that the immune system is healthy. And how do we supercharge the immune system? That is that it's gonna be definitely it's gonna be a combination therapies and putting our heads together. There's no other way.
In that regard, I completely agree with you. And the world of diagnostics, I have some experience with prostate cancer, and it's something really interesting. And I see even asking Holger's Dr. Heyn's question now. So now we're gonna use it, soon where it's gonna be approved, and we're gonna use it. And one of the things I wanted to get, it's just PBMCs from the patients before and after, just to see if the patients who have the greater response and longer response are the ones that you are generating quite like an antigen spreading, are the ones who have a better immune system. So the immune system, the radiotherapy generates change the environment enough so the immune system can get activated, and that response can get out from the tumor. And also I see another field of the diagnostics that I like, that is to target the bad cells in the immune system. The immune systems are well studied. We know that they have a lot of surface markers that sometimes they are specific from cell populations, and probably you could get rid of Tregs or MDSCs, because you have this marker for M2s, and you just and in fact, I think there's some work done now with FAB, just to kill directly fibroblasts associated with cancer. There's several diagnostic tools that being developed in that field, and I think it's not only targeting directly the tumor cell, to try to make it change and reshape the microenvironment of the tumor, but also try to reshape it just targeting a specific cell populations.
Absolutely, and I feel that this is something where we need to recruit a new generation of students, of aspiring oncologists, or people that really care about this, because there's no other way. The possibilities are endless, right? We have hundreds of cancers that we need to deal with. In diagnostics now, we have a new biomarker, fibroblast activating protein. We're destroying the fibroblasts that the cancer had converted into these zombies to protect the actual cancer from our therapies. So we have a wall of zombie fibroblasts that make up to 90% of the tumor. In the past, we were treating prostate cancer or breast cancer or melanoma, but we were actually dealing with that wall of protection from the immune system, right? And as we learn all these things, we have to recruit new people. We need to bring, just like the cancer is recruiting those cancer-associated fibroblasts, we need a new generation that embrace this. And I can tell you the future is bright. If you want an exciting future, get into this field and try to touch patients. And that's all. A life with a purpose is an amazing life. And I think we're all here enjoying what we're doing just because we can see the results in patients.
I think our audience will have got a lot of insights from this discussion. And as we approach the top of the hour, we can start to wrap up, although my feeling is that we're going to have to repeat this session to allow you to go in deeper, because there's such amazing synergies between all of you. So just echoing a few of the observations on the future. The future is bright. The future is collaboration. Science is we have to all come together. We need to embrace change and prepare indeed for the future, whether that is samples, embracing technologies, so on and so forth. And many elements, including not just recruiting the immune system, understanding the immune system, but actually using the immune system itself as a therapeutic, as some of you have mentioned as well. And ultimately, one of the most important elements I hear is really understanding our patients. So I, first of all, on behalf of our audience, really want to thank all of you, not just for your intellectual devotion to overcoming cancer, but the incredible dedication and humanity that you represent by focusing on patients. And I would like to bring our session to a close by asking each one of you for just one sentence for those audience members who are aspiring medical practitioners, oncologists, and technologists. If you can give your parting thought, what is it that they should do? And let's start off with Dr. Almaguel and thus bring our session to a close for today.
One sentence. You can be part of the change.
Dr. Prat.
Yeah, one sentence is basically join the revolution.
Dr. Piulats.
I would say, instead of a sentence, I would say 2 words, no? Hope and dream.
And I would agree that the future is bright, but the future is now. Just have to be smart enough executing on the data that we're generating today.
Thank you so much for your very meaningful words and your time. And I look forward to our continued discussions in the future. Thank you.
Thank you.
You're welcome. Great day, guys.
Yeah. Thank you, everyone.
Lovely.